thanks very much for sitting down with me today. I appreciate it.
Sure, you're welcome.
Potatoes have taken a little bit of bad press over the last number of years. You've been doing a lot of work on potatoes.
Mm-hmm.
Can you share with us the research of what you're working on, and what does a map genome mean for the potato industry?
I think I'm going to take you up on the first thing first, the bad press. I think that's pretty undeserved for potatoes. It is the third biggest global food crop, and it's actually very nutritious. It grows in a lot of different areas of the world, latitudes and altitudes. It's actually low on water input compared to other crops like wheat and rice. So I think the bad press is really undeserved, and often the bad press comes with what we have with the potatoes, the sour creams and the frying and the whatever. Yeah, so that, just to put the record straight.
Yeah.
So the research I've been doing is varied, and there's a multitude of different things I could talk about. Disease resistance is one of the things that I've been working on, but I think one of the most important ones that I'm still working on is called cold-induced sweetening. And what that is that for the processing industry, so the people that make the fries and the chips, they need to store their potatoes to have year-round production in the factories. And they do that at lower temperatures to avoid diseases, etc. But when you store potatoes at too low temperatures, the starches turn into sugars. And the sugars get an adverse reaction when you fry, and you get nasty brown chips that don't taste very nice. So I've been working on trying to solve that problem, to see, can we find anything in the genes in the potatoes that tell us that this potato is not going to do that in cold storage versus that one is. And so to improve the storage of potatoes. Actually, we have a collaboration project with Agriculture Agri-Food Canada to work on this as well. And we're trying to develop indicators that can, at the beginning of the season, when we store the tubers after harvest, to say, yes, this is going to be a good crop and we can store it for a long time, or maybe this has to come out earlier.
Right.
Yeah.
And so that mapped genome, what does that then mean to that type of research?
Yeah, the map genome, I think it's a start, it's a baseline. So the genome that was sequenced, and we were part of that, it was a huge international effort. It was something like 14 countries and 21 institutions, so it's a real global effort. We sequenced something that was a bit simpler than the regular potato. The potatoes that we see in the shop are usually tetraploid, which means they have 4 copies of everything, and it makes it very complicated to map the genome. And so we went to a very basic line that was not really one that you would grow for commercial purposes, but it gave us a very good baseline to work from. And once we had the baseline, we could do more, and we could develop additional tools. We now compare the potatoes that we grow and eat to that particular baseline, and we learn an awful lot from that. It's really, really, really advancing research very rapidly.
And I would imagine that taking collaboration to a whole new level.
Absolutely, yes.
Can you speak a little bit about that collaboration that happens between public and private institutions all over the globe, in the case of the mapping of the genome? Is there enough? Do we need to have more of it? And what should the industry be doing to foster more of that innovation?
I think it depends a little bit where you are in the world. Often industries are doing their own research, and when they do, they contract researchers to do the more edgy stuff where they do not have the resources as yet, where they're trying to figure out, should we put the resources there? And sometimes it's true collaborations where there is public breeding programs that then deliver material that maybe needs a bit more polishing before it goes out as a cultivar in companies. So it depends a little bit where you are in the world. I think there are very good collaborations in general. Could there be more? As a researcher, you always like to do more because it actually brings in the dollars that makes it possible for you to do your research. But I think in general we strike the balance pretty well.
In your opinion, why is it important to bring a wide scope of experience to an event like we're at the 13th annual SolGenomics Conference here at UC Davis?
So the Solanaceae Genomics workshop, the Solanaceae is a nightshade family, and there's a lot of economically important species in there. There's tomato, potato, pepper, eggplant, tobacco, petunia. They're all Solanaceae species, all nightshade family. And the more we find out about them, the more we find that there are similarities as well. And so we learn from each other, from the tools that we develop in the different crop species and in the different systems. I mean, some species are diploid, like humans, they have 2 copies of everything. Others are tetraploid, they have 4 copies of everything. Some can be inbred, some need to cross to other species, to other lines. So, you know, there's a lot of differences as well, but we use the commonalities very much to our advantage. So the annual meeting is to catch up with where the work is at, to learn from others, but also to talk to your collaborators. Like I said before, we have a collaboration with Canada. This is me talking from the New Zealand Institute of Plant and Food Research. And so this is a great way to have a project meeting. And Skype only goes so far. So it's good to sit face to face and have a good chat.
So you're saying that we still need to actually come together as human beings?
Absolutely.
Excellent. Very well.
I think so.
So as a private researcher, which sets you apart from a large portion of your colleagues that are here today, what do you see in the pipeline that's going to have the potential to make the most impact?
Well, I think the increasing knowledge of the genome, the genome mapping, will really help us. And so one of the things I can see coming very soon is the gene editing. Well, it's a bit controversial. Is it a GMO? Is it not a GMO? And different constituencies think about that differently. And I think most countries in the world haven't quite decided whether it is or isn't yet. But that means that we can go in and in a very targeted way make a tiny little change that can have great effects like disease resistance, drought tolerance, etc. And that's where all that knowledge that we gather from all the other species is very useful as well. Right. So that's one thing. And there's also examples of real GMOs like the innate potatoes that come out of Simplot in the United States. That's a GMO potato that's now grown in the States, and the products, the fries, are going worldwide anytime soon, I hope. Another thing is, and a development in potatoes is looking at diploid breeding. So that's breeding at a level where you have 2 copies of everything. It makes the genetics so much easier. And so there are a number of breeding companies that work on that, but there's specifically a company called Syngenta in the Netherlands who's trying to develop hybrid breeding, which is very similar to what's happening in tomato. And their research is progressing really well, and their results are really, really promising. So that would be something for the future as well.
Excellent. Thanks very much for sharing your insights today. I appreciate it.
Sure. You're welcome.